Okay. Um, well, thank you all for especially inviting me. I'm very flattered to be included in this workshop. Um, just a little bit of background on me. I'm an entomologist by trade. All of my professional training was in entomology, and then during my PhD program, I started working with viruses in grapes, with mealybugs in terms of vector ecology, and continue that on postdoc training at UC Davis with viruses. Then I took this post at the University of Florida to investigate palm infecting phytoplasma. So I apologize, in the title it says phytoplasmas in the Caribbean, but specifically I'm interested in the group 4 phytoplasmas, the palm infecting phytoplasmas of the Caribbean. Uh, but because of my training and what I do, I'm kind of a multidisciplinary research program where I investigate the insect vector ecology and management, as well as the plant pathology aspects with regard to the phytoplasma, everything from diagnostics to um, molecular biology. Let me dive into it. So again, I'd like to uh, thank you for the invitations for the talk. Just a quick thank you to the funding agencies that have supported my program and allowed me to make significant progress in this area of research. Um, so a little bit of background on the phytoplasmas that I work on. I came from a virology background, so I've kind of learned, uh, had some on-the-job training and learning with regards to phytoplasmas. But specifically, the group I'm interested in is the group 4 phytoplasmas. Now, you can see here that there are 4 currently recognized strains that belong to this group, subgroup A, B, D, and E. Uh, so subgroup A is commonly called lethal yellowing in the Caribbean, and it's the most predominant and widespread subgroup of this group of phytoplasmas. Um, and you can see on this map here that this is the geographic range of the group 4 phytoplasmas. Now, each of these marks on the map uh, correspond to subgroup A infections, but subgroup B is known from Mexico and the Yucatan Peninsula, as well as Honduras. Uh, Subgroup D is currently one of the more, it's the emerging issue today in Florida, and it's also known from Mexico as well as Antigua and Barbuda in the northeastern Caribbean. And subgroup E is kind of an obscure one that was documented from the Dominican Republic. Now, as I've gotten comfortable with, well, I guess as comfortable as you can be with phytoplasma taxonomy and how different groups and subgroups are named and established, and with some of the analyses I've conducted, um, historically they had subgroup C as part of the group for phytoplasmas. But as we're even using just the 16S gene, when this is analyzed, it doesn't resolve well with the other group for phytoplasmas, which is uh, not a big surprise given that it is known from coconut palms in Africa. So you have these two very geographically isolated areas where you have phytoplasmas infecting coconut palms in both of these areas. So I think in the early attempts and days of the phytoplasma taxonomy, because we had a case of what appeared to be convergent evolution in terms of disease symptomology, they were lumped into the group 4 phytoplasmas simply because they did they produce the same symptoms in infected coconut palms, where you have inflorescence, necrosis, premature fruit drop, yellowing of the fronds, and eventual palm death. However, when you look at them closely at the molecular level, it's pretty evident that they don't fit well with these other subgroups. We've just completed a study where we're looking at multiple loci, including the IGS region between 16S and 23S, GROEL, SEC-A, and SEC-Y, and doing a multi-locus analysis on all of these group 4 phytoplasmas. And consistently, subgroup C resolves well outside of the group 4 phytoplasmas. Now, because I'm not a real practice in phytoplasma taxonomy and the proper protocols, I don't know if the subgroup C, right now I don't know if it should be its own new taxonomic group or if it does resolve with one of the other newer phytoplasma groups that have been discovered. So that's something that I'm still not sure on, but what's pretty evident both from the molecular level and just the geographic distribution is that subgroup C doesn't really fit well with these other phytoplasmas. There was another report of a subgroup F in Florida, but recently we've found that it's not a valid subgroup. And one of the previous presentations, it was very exciting, where you mentioned that there was well-documented variability in the copies of these two regions within the genome, because what we found is this subgroup F was proposed based on RFLP analysis of 16S, and it looked like a distinct subgroup. So they proposed it, but when you look at the IGS region that is significantly more variable than 16S, it's 100% identical between what they've called subgroup F and subgroup A. So what I think is that the isolate of subgroup F that they called in Florida was actually just subgroup A, and there was some slight variability in the 16S gene between the two copies, and that's what was detected. So to recap, there are 4 legitimate subgroups of phytoplasma within the palm infecting phytoplasmas. Now, as I go forward, I thoroughly enjoy plant pathology and dealing with the pathogens, but by the nature of my job responsibilities and also passion for insects, this variability in distribution of phytoplasmas is exceptionally interesting for me within the context of the evolutionary relationship with vectors. So to go into detail on the symptoms that we see from these phytoplasmas, regardless of which subgroup it is, the symptoms are consistent among the different subgroups, where the initial symptoms that you're going to observe are premature fruit drop or inflorescence necrosis. Now, in a species like coconut, where it's always producing fruit, that's a very reliable character or symptom. But in some of the other ornamental species that have a more temporal production of inflorescence and flowers, or excuse me, and fruit, it's not always reliable. It just depends on the time of year. However, assuming that you can observe the necrosis of the inflorescence, the next stage of symptoms that progress are the discoloration of the oldest leaves in the canopy. And this results in a distinct layer of necrotic leaf tissue that's closest to the ground. And then over time, this discoloration and leaf death progresses further up into the canopy, affecting younger and younger leaves, until eventually you get the spear leaf collapse and palm death. So you can see images here on the left. There's necrotic inflorescence from a coconut palm. Then another image of a coconut. And if you look close to the crown near the trunk on the left side, you can see the collapsed spear leaf. And then on the right is our native cabbage palm we have in Florida that is exhibiting characteristic symptoms of what we're calling lethal bronzing, but it just represents subgroup D. But as I'm learning more about this group of phytoplasmas, the actual color of the symptomatic leaves is not terribly relevant, because you get different hues of discolored leaf in different palm species under different conditions. So the utility of those colors and the disease name is, it's not 100% accurate, but it's somewhat informative. But it's more of the combination of these symptoms together and how they progress that is indicative of these phytoplasma symptoms. So as I mentioned, my main interest in this is with regard to their relationship to insect vectors. Now, in the 1980s, this insect here, Haplaxius crutus, which is a succeed plant hopper, was documented as a vector of lethal yellowing, the subgroup A phytoplasma. And recently, we've gotten evidence, both from our research here in Florida, but also studies out of Mexico, has found that it also transmits the subgroup D phytoplasma. And for a while, nobody was able to replicate these studies. So there was some confusion and doubt with regard to Haplaxius crutus being the vector. And on top of it, a lot of the early vector work was done before there was reliable molecular tools to confirm infections in plants. So the transmission work done with crutus in the late 70s, early 80s was based off of symptom development and using scanning electron micrograph photos of infected tissue, which is it's fine, but you can't establish what subgroup is from an SEM cross section. So it wasn't perfect, but it was what was available at the time. Now that we have some reliable molecular tools, we're really starting to see progression in terms of vector research of these phytoplasmas. And then here on the bottom, on the left, is a lethal yellowing infected palm, and on the right is a lethal bronzing infected palm. And I'm going to touch on the symptomology of these palms in a little bit, and discuss what I think might be happening. So here I wanted to present an image. This is the male on the left of Haplaxius crutus, and on the right you have the female. So there's a noticeable level of sexual dimorphism in this species, and this is generally common in a lot of plant hoppers, where the female is generally larger than the males. And in this tribe of insect, the Ecoliini, generally you see a darker color in the females relative to the males. Now, when I started this position, our research focused here in Florida because we had active spread of this phytoplasma in our region. But it became apparent, once I got familiar with Florida and the local fauna, that invasive species are a common occurrence in the state because of our subtropical to tropical climate in southern Florida, and our proximity to the Caribbean and the Neotropics. So it's an ideal location for invasive insects to get established and possibly bring with them plant pathogens. So historically, lethal yellowing was introduced to the state around like the late. So initially, it was published in the early 1950s, but there's uh, accounts of it being here a lot sooner than that. But then uh, what we found was lethal bronzing was introduced to the state around 2006. And we conducted a study recently that's currently under review, and we found evidence that the local population of Hypaxius crudus in Florida is not genetically homogeneous. We've found four distinct haplotypes,
Yeah, that
and
yeah.
the predominant haplotype is identical to what we found in Jamaica. So lethal yellowing was originally discovered in Jamaica, as was Hypaxius crudus. So with the timing of everything, we suspect that both lethal yellowing and Hypaxius crudus were brought around the same time and allowed for the introduction of lethal yellowing into the state. Uh, Also, we do believe that a population of Hypaxius crudus was
I
brought
will
in
note that. Yeah,
um, and brought the subgroup D phytoplasma around 2005, and that caused our current outbreak that we're dealing with of the subgroup D phytoplasma. So, um, just some interesting, population genetics and biology of this insect that we're dealing with in the state. But it was always presumed that this insect was a native in Florida, but we're not so sure of that anymore. Now, one of the other things that I started doing with my job here was knowing that there's other species out there in the tropics on palms, and it's a poorly studied group, especially on palms. We initiated a survey throughout the Caribbean where we're basically going around collecting plant hoppers off of palms from different countries. So right now I have an ongoing project in Costa Rica. I'm working with researchers in Mexico, uh, Jamaica, Antigua and Barbuda, and Colombia. And basically, we've just been investigating the diversity of plant hoppers on palms throughout the region. And what we're finding is there is an enormous diversity of undiscovered and undescribed species of insect on palms throughout the region. And the exact number I don't recall off the top of my head, but we've described around 10 new species of plant hopper, a few from the Succidae and a few from Derbidae, that were all collected on palms throughout the region. So one of the things that I noticed and has made me concerned and interested in the vector relationship to this is that there are a large number of species that look very similar to Hypaxius crudus. This image here is a cross section of some of the succeeds I've been working with. On the left is Hypaxius crudus, which we've been discussing. Then to the right, you have Chamaceae, which is found from some of the smaller Caribbean islands. Hypaxius nidai, which is known from the Yucatan Peninsula. Hypaxius scarfion, which appears to have a wide range throughout Central America. Then we have a new species here to the right that actually last night I got word that the manuscript was accepted for describing this species, and it's going to be called Hypaxius pocococo. It was found on coconut palms, and then to the right of it is a recently described species we found named Hypaxius dougwalshi, and this was found on native palms, and it looks a little bit different, more different than Hypaxius crudus, but you can see that there's, it's easy to spot some of these differences using a microscope, but when you're in the field and doing large captures and transmission assays with large groups of insects, it's easy to see how you could miss and get another species mixed in with your batch. So it kind of highlights the care that's needed to do vector transmission assays in this pathosystem. And historically, it's been an issue just because palms always die when they get infected by this. And there's no known way to maintain a source of the group 4 phytoplasmas in a greenhouse setting. I miss the days of doing virus work when I can have an infected plant in my greenhouse indefinitely and at my own leisure do transmission experiments. With these palm infecting phytoplasmas, we don't have that luxury. I understand why people have had to, when they find declining palms, they run out, collect as many bugs as they can, and put them on recipient plants and hope to get transmission. We're at the mercy of what we have available, and it's not optimal in terms of understanding the finer mechanics of pathogen transmission. But that's what we're stuck with, with regard to transmission assays. So this series of insects here is just to try and demonstrate that there's a high diversity of closely related insects on palms, and I suspect that each subgroup of phytoplasma can be transmitted by multiple closely related species of insect, and a single species of insect in this group, I suspect, is capable of transmitting multiple, if not all, subgroups of the group 4 phytoplasma. But like I said, now that we're starting to understand more on the biology of the pathogen, we can start refining our methods. So that's what I want to kind of get into next.